Hello, I'm Rachel Deer, host of today's program, COVID-19, Keeping Up with a Moving Target. Thank you for joining us. This activity is jointly provided by the Postgraduate Institute for Medicine, DKB Med, and the Institute for Johns Hopkins Nursing. Today's program is accredited for ANCC, AAPA, and AMA PRA Category 1 credit. Please visit our website for complete CE information. If you're tuning into our webcast, please click the Claim Credit button on the webinar console. Otherwise, please go to covid19.dkbmed.com, navigate to our multi-specialty episodes, and select this webinar to claim credit. Today's learning objective is to discuss the current knowledge of the Omicron variant. This educational activity is supported by independent medical education grants from Gilead Sciences, as well as in-kind support from DKB Med. With us today, we have Dr. Paul Auwaerter, Clinical Director of the Division of Infectious Diseases at Johns Hopkins School of Medicine. Dr. Auwaerter, welcome back.
Thank you, Rachel. It's been a little less than a week at the time of this recording where we've learned of the latest variant that has generated a significant interest called Omicron, which the World Health Organization has labeled a variant of concern. And it's labeled it a variant of concern, perhaps the earliest of any variant, just several weeks after the first detection amongst infected individuals. It's actually a fascinating story that this variant has been described in such a short period of time, just 2 to 3 weeks, really arising from a respiratory outbreak amongst university students who were in the Johannesburg area of South Africa, but were traveling to the Western Cape of South Africa for a trip and became ill with a respiratory process and had genetic testing. Now, this was all done in mid-November, so just at the time of our recording here today, just under 3 weeks ago. And in less than 3 weeks, there has been this detection of a new variant. And how did that come to be? Well, the reason for this is some genetic platforms, molecular platforms that detect the SARS-CoV-2 virus uses PCR primers, such as this diagram here for the Thermo Fisher TACMAN kit. And there you have the open reading frame. You see there the spike protein and the N protein. So 3 genes, but if there is a significant mutation, the primers will not pick up the spike protein. This is called S dropout or S target gene failure. And this is what happened with the Omicron variant. And the reason South Africa was sensitive to this is this was also true for the Beta variant earlier. So once this group was identified as having the S gene dropout, the virus was sequenced and therefore the Omicron variant was described and came to the attention. Now, just having a new virus variant mutations, of course, why did this become just such a big deal? And the reason is if you go to where these students came from, which is the Gauteng province in South Africa. This is outside Pretoria and Johannesburg. In a very short period of time, in these past few weeks, 90% of the isolates taken from there are the Omicron variant. Now, this may be because it's quite a younger population. Illnesses have been generally described as mild. There's been no clear uptick in hospitalizations, although it's quite early. It's also important to state that the percentage of people immunized in South Africa stands at 24%, which is low, and this is despite the availability of vaccines there. So there's been some issues with uptake, and this has also been true worldwide, of course, with younger populations. There have also been reports that sampling of wastewater looking for the SARS-CoV-2 RNA has shown levels in wastewater very similar to the large Delta surge this past spring in the country, which might suggest that there's much more virus than health officials think right now that's circulating in the South African population. Now, what's gotten scientists and public health officials very concerned is this new variant has over 50 mutations uh, that have accumulated in developing this particular variant. And the bulk of these are in the spike protein. Of course, the spike protein is the component of the virus that docks to the host cells, the ACE2 receptor, but it's also the same protein that typically neutralizing antibodies are generated against and could render virus non-infectious or to help quell infection. So the Omicron is sort of the greatest hits of four prior variants that have all had their time in the sun, including Alpha, which was problematic last winter, Beta, which was a problem particularly in South Africa, Gamma in South America, and of course Delta, which is now the predominant virus circulating worldwide. This is a nice diagram from the Financial Times, one of the best that I've seen, that sort of catalogs these. And I won't go over the details, but for those who are interested, may want to pause here and just read over to get some sense. But largely, the mutations in the receptor binding domain of the spike protein and also the nucleoprotein both may cause problems in terms of existing antibodies not binding as well to this variant. And secondly, some of the nucleocapsid mutations may also increase transmissibility. And there's some very preliminary evidence that this could be the case. Again, numbers are very small, and you'll see this shortly in the following slides as well. But looking at the time that it took for a variant to account for at least 1% of the isolated virus in clinical samples in South Africa, you see the Omicron denoted here in red rose much faster than Beta or Delta, which is now the predominant virus in South Africa. So this suggests that it might be more transmissible than earlier variants. Now, if you look at the country itself, the numbers are still very small. What's gotten people concerned is that very low numbers have spiked upward to 3,000 to 4,000 coronavirus cases rather rapidly. And so numbers are by no means as large as earlier surges in the country, but you get the sense here that already Delta has waned. And at least for South Africa, the concern is that Omicron may be the predominant virus moving ahead in what seems to be another wave. So there are many unknowns, but the virus has already been described actually from an earlier sample in November in Botswana, Hong Kong, Canada, United Kingdom, Belgium, no doubt in other countries not yet understood. Transmissibility remains a question, as does disease severity. Sometimes viruses will mutate to become more successful, and that's by perhaps being more easily transmitted, but perhaps with milder illness. We just don't know enough yet. And of course, the big question on many people's minds are, is there some impact on vaccine-induced neutralizing antibodies or those that have arisen from natural immunity? So much more to be understood in terms of timing. Obviously, the experiments that will help try to answer some of these questions are underway, and perhaps by mid-December of 2021, we'll have some further information as well as some of the epidemiology that can give us some clues and insights into this particular variant. So Rachel, I understand we have some questions.
Yes, we do. And here's our first question. Do you think the rapid antigen test can detect Omicron variant?
So this is a very good question. Of course, one of the concerns with any variant that has significant mutations are, will it be detected either by molecular platforms or by some of the rapid antigen tests. First, I'll just answer for the molecular platforms. There have been some that have issues, such as I just showed you the Thermo Fisher platform that has the S gene dropout, but this is also something that's a important target to understand that you may have the Omicron variant without doing sequencing. The Roche platform has been said to be okay. So anyone who is ordering tests or in charge of laboratories certainly should await their manufacturers to see if their molecular platforms are at all affected by the mutations. And even if they are, I'm certain that new primers will be generated. This could be true just for single molecular platform as well as multiplex ones as well. In terms of the antigen tests, those, again, I've not seen any data. So until there's a sufficient virus that we can cultivate and analyze, we'll have to wait to see if this is indeed affected.
Thank you very much. Next question. Will remdesivir and monoclonal antibodies still be active against this variant?
So remdesivir is a nucleoside analog that interrupts the RNA genomic generation. And so that should not be affected by the variant. Monoclonal antibodies do have that potential. There's some concern, for example, for casirivimab and indivimab, one of the more popular of the monoclonal antibodies for treating or preventing the development of SARS-CoV-2 infection, because some of the mutations potentially affect the epitopes that are where the antibodies bind. But we'll have to wait for the experiments to be done, which should not take a vast amount of time, but several weeks, mainly because the so-called pseudovirus has to be made. And this is an important component of then analyzing the antibodies and whether it will have an impact or not.
And our last and very timely question. What are your recommendations about holiday and travel plans, particularly in light of this variant?
Well, I think in light of the variant at the moment, it's not clear it's here in the United States. So I think we still are flying by Delta rules, as it were. So until we understand more about the variant, I don't think it impacts anyone's particular plans. However, certainly there have been abundant numbers of countries that have experienced very rapid and high increases in COVID infections, particularly in Europe. And whether this will happen in the United States is not clear, but even within the United States, there are certain states, regardless of whether people have low or high immunization rates, that are experiencing upticks in infection. For example, Vermont, which has one of the highest rates of immunization, has had a substantial percentage increase in infections, as had other states, including New Mexico and Michigan. So a lot just depends on where you're visiting, whom you're visiting with, um, and what's your own health status. And I think this is the all the things I weigh when I discuss things with patients. If patients, for example, have significant health problems, or they haven't reacted well, or have health problems that cause them to not likely have a good response to vaccines, those are people that need to be more cautious, and especially want to make sure, if you're visiting other people, that they're either immunized or they take good care. I had a colleague tell me from London that the rates of infection there amongst young people and mask compliance is very low. Therefore, he wasn't recommending anyone going out to restaurants or indoors in public spaces, just because your chances of being exposed are very high. So again, this is something that has to be quite individualized. I suggest talking to your doctor that may have some sense of your own health risks if you're of concern, and then look where you're going and whom you're staying with.
Thank you for those updates, Dr. Auwaerter. If you're tuning into our webcast, please click the Claim Credit button in the webinar console to attest for credit. Otherwise, please visit us at covid19.dkbmed.com. Again, thank you for joining us and thank you for your dedication to your patients with COVID 19. Happy holidays and see you next year, Dr. Auwaerter.
And I certainly hope everyone stays safe and stays well during this holiday season. Best wishes to you all.